Hello Biology 182 students, my name is Jeff Mylander and I will be leading these online lectures for our hybrid version of Biology 182 here at Northern Arizona University. If you haven't already watched the How to Be Successful in Syllabus videos, please do so before watching this lecture as they will prepare you with the details of this course. E.O. Wilson writes in his book, The Social Conquest of the Earth, that in order to truly understand humans, our consciousness and the world, we must bring in other social conquerors of the planet like bees, ants, or termites. He writes, biologists have turned with great success to the bacteria and yeasts to learn the principles of human molecular genetics. They have depended on roundworms and mollusks to learn the basis for our own neural organization and memory. And fruit flies have taught us a great deal about the development of human embryos. We have no less to learn from the social insects, in this case to add background to the origin and meaning of humanity. This course is a survey of the organization of life, and you will be learning about those organisms mentioned by E.O. Wilson, among many others on this planet. You will also be learning form and function of many structural adaptations, how these organisms obtain energy, defend themselves, reproduce, the roles they play in their environment and our economy, and much more. This is just an example of some of the specimens you will be studying this semester. On the left is a color-coded key that shows you the taxonomic levels you will become very familiar with. This image represents the algae, plants, and fungi you will be learning, and this image shows you many of the animals you will get to know. As a side note, your lecture and lab will not be aligned as far as the material you will be studying. In lab, you will be focused more on the taxonomy and traits of specific organisms. We'll start our discussion with systematics. To begin, we can see a family tree on the left, something you might be familiar with from past classes. And on the right is a cladogram, something you might be familiar with if you have studied biology before. If this is the first time you have ever seen this, don't worry, it won't be your last. We will spend plenty of time getting familiar with these and you will see them on tests, so start practicing now. Now looking at the cladogram, which organism evolved first, fish or marsupials? And how many years elapsed between their evolution? Remember, you can pause the video to give yourself a chance to think about the questions in these lectures. If you answered fish, you would be correct. But how do you know this? This is the important concept you need to grasp. Cladograms are associated with a timeline. Sometimes they are present, as you see in this image, running up and down the left side, and other times they aren't. But it is understood that the cladogram exists over time. As you can see here, the branch that leads to the fish extends to 400 million years ago, while the branch that leads to the marsupials only extends back to about 150 million years ago. Therefore, we have a difference of about 250 million years. So what is systematics? Well, to put it simply, it is the understanding of evolutionary history of life on Earth. There are three concepts that we will be studying to gain a better understanding of systematics. They are taxonomy, the identification, description, and naming of an organism. For example, humans are referred to as Homo sapiens. Classification, which is placing an organism into hierarchical groups to show its relationship to other organisms. You may have heard some of these groups before. Kingdom, phylum, or class are some examples. And last is phylogenetics. This is studying the evolutionary relationships between organisms using morphological and genetic data. We use cladograms and phylogenetic trees to help visualize these relationships. So let's take a closer look into each of these topics. How do scientists name organisms? Well, the correct answer is binomial nomenclature. Binomial nomenclature was created by Carolus Linnaeus in order to have a unifying naming system that worked across all languages. How about scientific names? How do we write them? There are three rules, and they are as follows. First, you capitalize the genus. Second, you do not capitalize the specific epithet. Now, you might be asking yourself, what is a specific epithet? Isn't that the species? Many people have incorrectly learned that the last part of a scientific name is called the species. The species name actually consists of the genus and specific epithet together. And last, if you are writing the name, you will underline it, and if you are typing it, you will italicize it. Here are some examples. Humans are referred to as Homo sapiens. Homo is the genus, sapiens is the specific epithet. Homo sapiens is the scientific name or the species name. Saguaro cacti are referred to as Carnegie gigantea and were named after Andrew Carnegie, a very successful philanthropist and industrialist. And polar bears are referred to as Ursus maritimus. Our second concept is classification. Here, scientists place organisms into 7 taxonomic groups. So what are those taxa? Well, you may have learned all these in previous classes, but they are kingdom, phylum, class, order, family, genus, and specific epithet. They are listed from the broadest category to the most specific. There are many mnemonic devices to remember this, but kings play cards on fat green stools is a good one. Last, we have phylogenetics. Do you remember what a cladogram is? They're also called phylogenetic trees, and they show evolutionary relationships between organisms. Here's that example we saw earlier in this presentation that represents several vertebrates. We call this orange dot a node, and it represents a common ancestor between 2 groups or organisms. In this case, natural selection acts on 2 different populations of a common ancestor, creating 2 distinct lineages. One evolved into what we see today as birds, and the other, as we can see here, led to another common ancestor, acted on by natural selection, which led to monotremes and eventually mammals. Here are 2 terms you will need to become very familiar with: ancestral and derived traits. What do you think the difference is between the 2? An ancestral trait is one that evolved before the common ancestor of a group being discussed or considered. Think back to that orange dot we called a node. That was a common ancestor. So an ancestral trait would have existed before that on the cladogram. A derived trait, on the other hand, is a trait that appears for the first time in a group which defines that group. So practice really quick. What might be an ancestral trait of primates? Remember, it would have existed before its common ancestor. Hair would be a great example of this. We see this in earlier evolved animals such as mice. How about a derived trait of primates? If you said opposable thumbs, that would be an excellent answer. Opposable thumbs have not been seen before the primates evolved. Here's another opportunity to practice more with cladograms. Answer the following questions. Okay, so hair is a unifying trait for which organisms? The correct answer would be horse, wolf, leopard, cat, or you could refer to them as class Mammalia. Number 2, the most recently evolved organism is the domestic cat. Remember your timeline that is associated with cladograms? It's just not present here. 3, the wolf, horse, and turtle lack claws. And number 4, the wolf, leopard, and cat are carnivores. They belong to order Carnivora. And our last question, which type of trait would be represented by these orange boxes on the cladogram? These would be derived traits because they are specific to the horse or the wolf. They are located on the branches leading to those organisms, meaning they are specific to that organism, or if you were looking at a group of organisms. You can also see that they are located after the common ancestor of that organism. Cladograms can be tricky, so it's best to practice these each week. Place all of the phyla, subphyla, or classes you learn each week on a cladogram and add ancestral and derived traits as you learn them. You will need to know how to build a cladogram as well as read one for your weekly tests. 2 other terms you will need to be familiar with are homologous and analogous traits. A homologous trait is shared by 2 or more species that has been inherited from a common ancestor, but may not always look the same because it generally is modified to perform a certain function. In this example, you can see that the bat, mouse, and human all have the same bones in their upper extremities, a humerus, radius, ulna, carpals, metacarpals, and phalanges, yet they are modified to perform different functions in those organisms. An analogous trait is a trait that is shared by 2 or more species and serves a similar function but is not derived from a common ancestor. In this case, the ability to fly was evolved by mammals, birds, and insects at different times throughout evolutionary history. Analogous traits can be tricky. Don't get confused, as they don't always represent a similar ancestry. When this happens, we call it convergent evolution. This is the independent evolution of similar traits because organisms may experience similar environmental pressures and therefore evolve similar adaptations. Now you might be asking what I should study for the first test. Make sure you understand and can define each of these concepts. Here are more questions for you to practice at home. Once you answer these, try making your own cladogram or look up some other examples online and make up your own questions to practice. At this point, you should read through the first activity packet over classification and taxonomy. In your first week of lab, you will be using specimens and working with your lab mates to complete activities 1 through 4. Our next section of this lecture is going over the microscopes. Some of you may have a lot of experience with microscopes and others may have little to no experience. Either way, you will be using these pieces of equipment every week, so you will need to know how to use them and find their specific parts. At your lab benches will be 2 types of microscopes. The first is a dissection scope, and the second is a compound scope. You will mostly be using the compound scope in this lab. They each have similar parts that you will need to identify and become familiar with. Dissecting scopes have a greater working distance and are used when you have to examine large or thick specimens and be able to manipulate them while you observe them. Compound scopes, on the other hand, are used for small, thin sections of organisms through which light can pass, for example, skin cells or leaf cross-sections or maybe spores. In lab, you will be using these scopes to view prepared slides or wet mount slides which you will make. This slide has links to YouTube videos that will teach you how to use these scopes so you are prepared before you show up to lab. Please click on the links in the PowerPoint that your lab TA has uploaded to BBLearn or search YouTube for the video titles listed in this presentation. Now that you have watched the videos on how to use a microscope, this may be a good time to pause the video and read through your Lab 1 microscopy reading packet and work on activities 1 and 2. When you get to lab, please be sure you can identify each part on the microscope because they differ from the ones you will see in the packet. You will need to complete activities 3 and 4 in lab. When you do begin working with the microscopes, they will already be at your lab tables. Once you uncover them, you will first adjust the oculars for your eyes. Make sure you use both eyes because there is a pointer in the right ocular that you can use to put on structures or specimens when you are asking questions, or the TA will use to identify questions on a test. Always start with the lowest power and work your way up. It is the red objective, also called the scanning objective, because you will be scanning your slide to find the specimen. Bring it into focus before you move to a more powerful objective. In this lab, we never use the oil immersion lens or the 100x objective. Once you have found the specimen or structure, brought it into clear focus using your coarse adjustment knob and then followed by your fine adjustment knob, and you get it to fill your field of view, you will want to calculate the total magnification by multiplying the magnification on the objective lens and the ocular magnification. For these microscopes, the ocular magnification is equal to 10 times. Last, before you make your drawings, you will want to adjust the light up or down. This can help give you a clearer image of the specimen or structure in question and will help you make a more precise drawing in your notebooks. Your lab TA will go over making a wet mount in lab, but we will quickly review it here. All the equipment you need should be at the center of your lab benches. Get a glass slide and add a drop of water to the center. Place a small piece of your specimen in the drop of water and then grab a cover slip. Lower it down at an angle over the drop of water and specimen so it reduces air bubbles in your view. Once you have done that, observe your specimen at all magnifications, except the oil immersion lens, in order to get a good understanding of what it looks like. Use your lab atlas as a reference tool to find the specimen that you're looking for. Choose the magnification that gives you the best view of the structures you are supposed to identify and label, and then make your drawing. Don't forget to label everything and record the magnification. So what is a field of view? The field of view is the amount of area seen through the microscope at each given magnification. Why might it be useful to be familiar with the field of view? Well, the field of view is important in order to understand the size of the specimen or structure you are investigating. Some microscopes are fitted with a tiny ruler, micrometer, inside of the ocular that will allow you to measure the width of your specimen like cells or their structures. When making drawings in your notebooks, fill your circles to represent exactly what it is you see in your field of view and do your best to draw everything to scale. This will help you develop a better understanding of the specimen and your spatial recognition on a test. If the specimen is really small, zoom in to a higher power. How does the field of view change as you increase in magnification? As you increase magnification, you are zooming into your specimen to get a closer look. Therefore, your field of view decreases as you increase magnification. For example, you can see with the penny, as we go from a low power here to a higher power, we get a closer look at Lincoln's head. You are seeing a smaller portion of the penny enlarged. Again, make sure that when you make your notebook drawings, you are zooming into a magnification that shows you all of the structures you need to label. How about depth of field? What does that mean? This is the portion of the specimen that is in sharp focus, essentially the topography of the specimen. How is it affected by increasing the magnification? The depth of field is important in order to understand the layering that occurs in your slide. As you move the fine focus back and forth, you are able to get a better understanding of the specimen you are looking at and create a 3D image. In this example, under low power, you can see all 3 strands of thread are in focus and you can see each one well. However, under high power, a much smaller depth of field, we can get a really good look at the red strands, but the yellow and blue strands are out of focus. It's similar to photography in that a higher f-stop gives you a deeper field of view and a lower f-stop gives you a shallower field of view, blurring the background. For your first week of lab, be prepared to turn in your signed syllabus contract. Ask your TA any questions about the lab setup. Communicate and clarify any disability-related concerns or any known university excused absences you have coming up. Finish your lab activity worksheets in lab, and when you are done, staple them into your notebook. You are not making any drawings for lab 1. If you have time in lab, start getting lab 2 set up in your notebooks. If you also have time in lab, you could start working on post-lab 1, so print that out and bring it with you to your first lab. And start preparing for the first test. Making flashcards is a great method. And last but not least, schedule a time to watch the Lab 2 Algae screencast. Good luck studying and getting accustomed to this lab. See you again in next week's lecture.